So good morning, everyone. Today my talk is going to look at overcoming the barriers of biophysics in the gene therapy space. As we all know, the gene therapy has exploded in the last few years and has become a very hot area in the pharmaceutical industries right now. It is also producing very transformative medicines. And I would like to really emphasize the role of the biophysics in pushing the boundaries of this gene therapy field. So before I go into the specifics of the biophysics and its role in the gene therapy field, we all know about the CQA of the AAV system, which can go from using mass spectrometry for whole cell proteins to post-translational modifications to a battery of different tests ranging from ddPCR for your titer levels, plasmid DNA, to your TCID50 assays. Usually biophysics is used for characterizing the distribution of your adeno-associated virus. In this case, you're looking at your percent empty capsids, percent full. In certain cases, you can look at your impurity profiles and even your DNA to virus particle loading. It can be used at times to look at process development changes, manufacturing consistency, and so forth. Other techniques such as DLS can look at your higher order capsids, in which this is defined as anything above your anything larger than your full capsids is going to be called your higher order capsids or aggregation. You can even get true extinction coefficients in certain cases of your empty, full, and larger capsid species. So right now I'm going to talk a little bit about the traditional biophysical approaches that we have been using to further advance the gene therapy field. For example, looking at your aggregates or HOC regions, DLS is used commonly to look to see as a qualitative assessment of your aggregates in your system. Remember, intrinsically, DLS is not a quantitative system. AUC, which I'll touch upon and focus on in the remainder of this talk, can also do that. Other size distributions for your low order capsid, which is anything smaller than your empty capsid, empty capsid, partial, full, and HOCs. You can use secmols, papillary, ISO, electric focusing, TEM, and cryo-EM. On the right hand side here, you see a nice chromatogram of the secmols, which gives you a good idea of your empty and full capsids. However, there's a limitation to each one of these techniques. In this case, you can't really get a good idea of your partial capsids. And then it's also dependent on the front end of the mols, which is your LC. So in that case, it's going to be your the limitations of your size exclusion chromatography column. Other approaches such as clever chromatography as ion exchange can give you good readouts for your empty and full. And again, it's all dependent on these type of affinity-based chromatography, and which has its problems. Another one is another type of techniques is the charge distribution mass spectrometry on the right-hand side here, which can give you a good idea of your empty, partial, and full. But again, this is limited to these particular region, higher order capsids, reversibility, and so forth. It's going to be very difficult to attain. Other specific chromographic methods, that BIA separations with these special columns, can also give you some good relative information, but again, suffer from some limitations. So the remainder of this talk, I'm going to talk about AUC. Specifically, it is a gold standard for particle characterization. All of the techniques that you have mentioned before come back to AUC as its proving ground. It compares itself to AUC, so why not just use the gold standard at all time? Now, AUC has been around for over 98 years and has been proven out throughout time. It's just right now it has played a prominent role in the gene therapy field. The FDA have seen it, the EMEA have seen it, and it's one of these critical parameters, for example, your percent empties, is being determined by AUC. It's also a very multifaceted technique that's based on first principles. So there's no type of dilutions, there's no type of matrices, there's no need for labeling or staining. It's compatible in many systems. both biological and non-biological, and it is very, very sensitive once performed correctly. You can have LODs, level detections less than 1%, and LOQs less than 1.5%. So it is a very robust technique. So not only can you do absolute quantitation without any type of empirical measurements, i.e. you don't use standards, you can do it as a function of just putting your sample in and doing the analysis. There's no need for any type of calibration or so forth. In addition to that, you have the hydrogen-like properties, which is very critical to your gene therapy system. So you have a very broad size distribution range that goes from sub-nanometers to microns in terms of you can look at a cell, a whole cell in the AUC. It's independent of any type of reaction kinetics, i.e. I'm talking about self-association, heteroassociation, and reversibility. You can get good, insert a good idea of your molecular weight in certain systems, the shape of the system, your hydrogen radius, which is kind of very complementary to the DLS data analyses, and you can, in certain cases, calculate your diffusion coefficients. Usually, AAVs are about 1.8 fix, which is pretty consistent between DLS and the AUC. In addition to that, there are limitations known to this, and these are the commonplace limitations which are most people refer to when talking about AUC. They are long experimental times, depending on your rotor speed. It can be overnight, 4 to 8 hours. It's expensive instrumentation, and the data analysis requires some level of expertise. Actually, you have to know what you're doing, because the programs that are used to solve for the equations that in terms give you the model of the data are complicated. And this is something that is plaguing the field, but I believe, and I'm seeing right now, a resurgence of this, of the AUC, which also correlates to a resurgence in the education. So this expert requirement is going to be phased out in a short time. Now, why do we choose AUC? So AUC can be used for a variety of different qualities of your system. So you can look at, we all know about your capsid quantization, the HOC characterization. We'll talk about this intracapsid heterogeneity a little bit in this particular talk. And this is complementary to your capillary isoelectric focusing. Actually, it's complementary and gives you much more data, but it just gives you the power, demonstrates the power of the AUC. Your genomic DNA capsid loading, you're going to get those ratios. You use this with a multi-signal type of sedimentation velocity experiment that we routinely use to determine stoichiometry of a heteroassociation. So this is commonplace type of experiments when you're looking at binding interactions. And obviously you can look at your contaminants. So your LOC again is anything below your empty capsid. So you can look at proteins that are tagging along with your viral particle, which I've seen a lot. Some of them could be Rep68. It could be any other VP1 type of aggregate or so forth. You can actually quantify that. And really assess what it is, even free DNA in there. So routinely we look at your capsid quantitation and the HOC characterization. In the upper right panel is basically your commonplace empty capsid quantitation that I spoke about briefly before. And then taking it and expanding upon this, you can do your HOC type of quantitation. Not only HOCs, but you can look at partial capsids and design methods that can really quantify not only your empty capsids and your full capsids, but also your partial capsids. And in certain cases, you can design a cGMP method to look at 2 analytical or 3 analytical responses under certain conditions. So to go into a little bit more about your intracapsid heterogeneity, what I'm going to talk about is simulated data based on real world examples. In this case, we have, I modeled an empty capsid at 10%, a full capsid species at 20%, a full capsid species at 30%, and a third capsid species at 40%. Now they're very similar in S values. So when we take this model data and we simulated this under these conditions of your routine characterization. What you get is basically two peaks. You get your empty capsid on the right-hand side for your CS distribution, and you get a peak of your full capsid. Your boundaries look bimodal, which indicates, it might be only two species in there. However, we know there's not two species. So at this point in time, this experimental design did not capture the heterogeneity in that full capsid that I was looking for. I did not model any intermediate capsids. I'm limited by that. So what we want to look at is how can we figure out these three species in that second bottom right graph, CFS distribution. I took the same data and modeled it in another program to show that you do have the three species in there, but we already know that a posteriori. So in this case, the data during the routine characterization did not really capture that capsid heterogeneity. However, if you change experimental conditions that favors a higher resolution, you can actually see this capsid heterogeneity within your system. On the lower left is that experimental conditions which produce the upper right hand CFS distribution, which in turns we know now that we do have the three species in there. You can use orthogonal methods such as sudden hybridization of your packaged DNA, in your full capsid, to really confirm this, which it has been done in my hands. So, or this type of analysis have been proven out by orthogonal methods. And this is another characteristic of the AUC that shows the utility and the versatility and the power of doing these type of analyses. I presented here the overlay of the original routine characterization with the optimized experimental conditions to capture the heterogeneity within that system. And you can see it overlays. So basically what we're doing here is increasing the resolution to get your data better. Now, you can fit each section differently and get pretty much the same thing. So this just demonstrates some of the power of the AUC. So let's go into some of the general characteristics and functions of the AUC and what it can do. So we can do for cGMP method development and validation of basically your QC release, stability, comparability, and process development. So what I want to really focus on is the cGMP method development and validation. Now, there's a couple of camps. There's two camps in the AUC world. One camp that says that this method is pretty complicated and the data analysis is very complicated and it can't really be translated into the cGMP world. And then there's a camp that I sit in which we have proven out that this AUC wants a robust and an emphasis on the method development that precedes the validation is focused on. You can get a QC release method that is going to be accepted by the FDA once done correctly, and it has been in certain cases. So as I said, I want to stress that you have to put a lot of emphasis on your method development. That comes from the experimental design. For example, if you're looking at a 1% type of empty capsid in there. If your loading concentration is 0.5 OD units, then 0.05 of it is going to be in your representative of your signal of your empty capsid. That gives you a very low signal to noise. So you have to design the experiment that will give you the best signal to noise analysis. In addition to that, you have to shore up your final analysis. And the way to shore up your final analysis is to explore the analytical landscape of it. What I mean by that is to do orthogonal modeling of the data to really, really shore up that final output that you're going to present as your validated method and as your validated system. So you have to take that into consideration and really put that emphasis on the method development before you even go into the validation protocol. If you don't, you will have OSs and deviations. Another thing you can do, we want to push the boundaries, and especially at my company, we want to push the boundaries of the cGMP method development. And what we do is we try to not only look at a single analytical response, but under certain conditions, you can have multiple analytical responses. So, for example, you can develop a method that not only quantitates the empty capsid, but it quantitates your partial. And if you're clever enough and your system is amenable to it, you can also do your aggregates in this case. So it's all very flexible, and under certain conditions, you can do multiple analytical responses in just one method. Now, a very critical part of it is the integration of your data. This is the meat and bones of the CFS distribution. So you have to have a system in which you have discrete species that define your empty. So in this case, you don't want to have species that are looking very close together, and the QC analysts have to make a choice. You want to remove the subjectivity out of the integration, and that comes from your original analyses. But also, it's the integration of it. For example, you don't want to use peak heights as your sedimentation coefficients. You want to use the weighted average across the peaks. That's a fundamental problem that I've been seeing. What you also want to do is have a good integration system, and we have developed a custom integration system that was developed by AUC users like myself to really focus on the AAV integration in terms of a cGMP environment. And we call this a capsid integrator. So that is unique to every method, and it's very amenable to the 21 CFR Part 11 compliant. It is very compliant. Okay, so another thing we can look at is the stability. So when you put your samples over a stability program, it's very good to use the sedimentation analytical centrifugation to look at a size distribution profile, because you will see the formation of your degradation products or formation of aggregates, and you can really do time 0 point to your 30 days or whatever you're doing your stability program at, and see and characterize those changes very robustly. So for example, if you're looking at dimerization of your system in terms of, or multimer of your system, you will be able to see that in the AUC. It just have to depend, it's very dependent on how you design the experiment to capture that. So that's one of the things you have to focus on is how these experimental designs can capture these type of degradation products. But it's SV-AUC is routinely used for stability studies. In addition to that, this is where the SV-AUC can really help your process development in terms of, you're making changes upstream and downstream. You're changing from a cesium chloride gradient to a AEX chromatography. You're doing all these changes. You're taking away certain processes, certain steps, or increasing certain dwell times or something like that. You can look and see the changes, and this aids in the de-risking strategies that you have and mitigation of those risks by looking at your process development samples as it's coming out in real time. Now, the experimental times is you say it's long, but you can do the experiments. I mean, at an 8-hole rotor, you can hold up to 7. I wouldn't recommend using 7 analyses at one time because of the size of these particles. But you can, in a week's time, you can run at least 15 samples in a 4-hole rotor and get really good data. Now it's going to take you about a week to figure out the analysis, a week to maybe 2 days to a week, depending how skilled you are. But at the end of the day, you still it's a quicker turnaround time than it's a quick turnaround time. Plus, it's a very informative data analysis and reporting of what you're going to get and see during your process development cycle. In addition to that, we also look at comparability from batch to batch. And I want to make sure that you're going beyond the empty capsids. You're looking at changes in your capsid regions. Is the peaks broadening? Are you seeing a shoulder appear? Are you seeing more of your HOCs? These are the comparability studies you can do to really see what happens from your batch to batch, your variance in your systems. But overall, the AUC has presented itself as one of the workhorses of the AAV field. And I know I stressed a lot about this in terms of the biophysical realm, but I think it's very important to really figure out what technique can help you in getting your product to market. And in this case, AUC is one of the best ones. So I think that's the end of my talk. So if you have any questions, please ask me.